Welcome to lab number 11. This is seed germination. And there are different ways this is done depending on our availability of seed and other materials for various types of germination testing. And so there's really not a whole lot of video required to describe what we need to do for this lab. If you are doing this in a face-to-face environment in the classroom, then you will already have a list of the treatments of germination, because we're going to try germinating seeds in at least two different ways, with a clear open germination that's exposed to light and germination that is exposed to complete darkness only in foil. So it's wrapped where no light can get to it. There are also different colors, different types of seed that we may or may not try. So read the lab. I'm not going to walk through and talk about all of this. There's really very little worth talking about additionally anyway, since I think the text here is done very well in combination what I've already talked to you about in class, or we'll talk about in class shortly, depending on when we do this lab. So read the vocabulary words, read the description. The way we do this is take little Petri dishes, little plastic dishes that we put some blotter paper inside, cut in little circles, and add water and seeds. And we can then take those seeds and germinate them under the appropriate conditions, properly labeled and properly dated with your group name and information on each container. And so table 11.1 allows you to record all of the data. What is your group doing? If you're given more than one crop per group, and what the treatment is. Is it going to be in bright light, dim light, colored light, dark, whatever? Dark would be foil wrapped, for example. So this is a treatment key that we've used in previous years, where there is a total possibility of 8 treatments, either clear, nothing covering the Petri dish, red, aluminum foil, or blue colored cellophane materials. And then that put in bright light. And then the clear low light, red low light, foil low light, and blue low light. So those are treatments that we've commonly done in the past, and I may or may not decide to do it differently face to face to look at 8 different setups. Again, with more than one seed type, usually I typically select at least 2 types of seed in the class, and we have to have enough students to do that many different treatments and all of the materials. And so here's where you can record your seed germination data. So T0, there's nothing germinated. So you just put in how many seeds that you used in your tray for the type of treatment, and then you would label it either low light or bright light. Low light I would just put inside of the fume hood in our classroom, where, yeah, it's not completely dark, but it's dim. It doesn't get a whole lot of light. When the classroom lights are on, it's certainly not dark in there, but it's definitely different than being in a sealed box that's completely dark. T1, 2, and 3 are the different times, depending on what kind of access time we have to read. Professionally, you might read them every day, or you might read them first time after 3 days, and then after 6 days, and after 9 days, or it depends on the species of plant that you're looking at. They all have different rates of germination and different appropriate times. So that's how you can record the data. Again, there's room for lots of data to be gathered. Making sure that you include some notes. What do you observe? Not just the raw numbers. Well, 6 seed were germinated out of 20, but were the seeds growing well or vigorously? Were they growing uniformly? Was there any color issues where they were very, very, very pale or yellowish versus green? Was there any mold or sliminess that might indicate bacterial growth in your trays? Anything you see. And then there's some room here in the lab to just put some general notes about the class as a whole. Anything that was a common notation that you wanted to note about the things that you observed. And so if we do it in class, you've got to fill all that in. If you don't do it in class, then you're only required to fill in the answers to these questions. So there are several questions on, I think it's 2 pages of questions, or a page and 2/3, because the top of page 1 there is for notes. And that's it. And you can look up the answers to those questions based on material presented in this class. You can look up information online and do your best to answer completely all of these questions that are on the pages 91 and 92 in your lab book.